Hi, my name is Andrew Kershaw. This is Tuesday, 12/13/2022. And today I'm going to go over my application activity number 4, the movement of macromolecules. All right, so my first macromolecule is a steroid hormone, cortisol. Cortisol is a specific type of steroid hormone classified as a glucocorticoid, important to the human body's ability to respond to stress by helping it prioritize energy stores throughout the body. Cortisol production is controlled by a biochemical pathway involving the hypothalamus, the pituitary, and the adrenal glands. This is also known as the HPA axis, where the hypothalamus produces a hormone called corticotropin releasing hormone, CHR here in my diagram, which is transported to the anterior pituitary corticotrope via a system called the hypothalamic pituitary portal system. Here, CHR will then bind to the receptors on specific cells within the pituitary gland. These receptors are called corticotropes. This will stimulate the pituitary gland to ultimately secrete adrenocorticotropic hormone. This is ATCH in my diagram here, right here. This will be secreted into the bloodstream. This will then travel down to the adrenal gland, where it will stimulate the actual production of cortisol from a region in the pancreas called zona fasciculata. All right. After the synthesis of cortisol, it will enter the bloodstream from the adrenal gland. Once in the bloodstream, about 90% of the cortisol created will actually bind to something called the corticosteroid binding globulins. However, the remaining free cortisol will permeate different cell types, so cells in the liver, muscles, the bones, adipose tissues, or neurons. And it will bind to something called a glucocorticoid receptor, this is GR in my diagram here, within these cells and promote the actual changes by cortisol. One example of the changes cortisol causes in the body is relating to the immune system. So, for example, once bound to these glucocorticoid receptors, this cortisol and GR complex will then move to the nucleus of the cell, where it can then bind to different transcription factors involved in the synthesis of inflammation proteins. This binding to that synthesis, to that transcription pathway, will actually inhibit the inflammatory protein's ability to actually be expressed and ultimately created. So that's one pathway cortisol can actually reduce inflammation in the body. The other way is it could actually, this same cortisol GR complex, once it's in the nucleus, it could also bind to transcription factors and promote anti-inflammatory cell signaling, which will create anti-inflammatory proteins that will basically mitigate the effects of any inflammatory proteins that have been released throughout the body. Cortisol will eventually return to the bloodstream via the process of exocytosis. Once in the bloodstream, cortisol can be converted into cortisone by an enzyme called 11B dehydrogenase, or cortisol can be metabolized in the liver or the kidney by enzymes 5-alpha or beta reductase, and then ultimately excreted in the urine or feces. So that's cortisol. So we can now move to my protein hormone, which is glucagon. So glucagon is a 29 amino acid peptide hormone secreted predominantly from alpha cells contained in a specific region of the pancreas called the islets of Langerhans. Glucagon's primary function is related to glucose homeostasis within the body. Specifically, glucagon production is triggered when the body is experiencing like a hypoglycemic condition. So when glucose is very low in your bloodstream, glucagon concentrations will increase, which will ultimately stimulate cells within the liver to produce glucose and increase glucose in the body to normal levels. Glucagon production starts with the expression of a precursor called proglucagon from within the pancreas, as well as this process also takes place in the intestines or the neurons. Proglucagon is processed by an enzyme called prohormone convertase 1 and 3, or prohormone convertase number 2. This is either PC1, 2, or 3 in my diagram here. Depending on the location of the proglucagon expression, a different PC enzyme will be used. So in the pancreas, proglucagon will be basically processed by PC2, turning proglucagon into glucagon. But in the intestines or in the brain, proglucagon will be processed by PC1 or 3. Once created, glucagon is packaged inside intracellular vesicles in the Golgi apparatus. Hypoglycemia and the corresponding low ATP levels within certain cells, specifically pancreatic alpha cells, will trigger a lower concentration of potassium within the cell. This reduction of potassium in the cell will actually open voltage-dependent calcium channels, allowing calcium ions to flow into the cell. This basically down gradient will allow the cell to basically bring in glucagon from the blood into the actual cell. So now glucagon is in the cell. From here, glucagon will enter the bloodstream and travel through the portal vein system into the liver, where it can bind to a receptor called the 7T receptor on liver cells, so hepatic cells. And this will actually initiate the production pathway for glucose within those liver cells. Glucagon clearance now is actually something that's still being actively studied. Therefore, the specifics of the degradation process for this hormone is not completely understood. However, glucagon degradation is believed to occur within the liver or the kidney. It's actually facilitated by receptor-mediated endocytosis, meaning from the research I was doing, it sounds like glucagon is actually brought into, it's basically processed within a kidney or liver cell. So it's brought into those cells and then broken down in the cytosol of those cells by an enzyme called dipeptidyl peptidase 4, and then it's ultimately excreted from those cells and excreted in the urine or feces. Okay, so that was my protein hormone glucagon. And last but not least, I'll talk about my intracellular protein, hemoglobin. So hemoglobin is an intracellular protein found within the cytosol of mature erythrocytes. This is just red blood cells, right? So hemoglobin's primary role within red blood cells is to bind free oxygen diffused in the lungs and transport it throughout the body. So hemoglobin synthesis tracks really with the maturation process of the red blood cell. So as the red blood cell is maturing, more and more hemoglobin is being created. So the synthesis of hemoglobin requires two things, and it's a coordinated process. It requires heme and globin proteins. Heme is a prosthetic group that mediates the reversible binding of oxygen in hemoglobin. So this is the actual substance that oxygen can bind to within hemoglobin. And then globin is this protein that the heme attaches to, and that actually creates this whole hemoglobin structure. So it actually protects the heme molecule. Heme is created in a pretty complex process, starting with succinyl CoA formed in the Krebs cycle, found in the mitochondria, and is then bound with glycine. From here, and I have this in my diagram, it goes through a number of steps. So you can see there's 1, 2, 3, 4, 5, 6 steps involving a number of different intermediates and enzymes, to where it is eventually turned into something called protoporphyrin 4, which is then combined with an iron atom here to create heme. And so this process takes place in both the mitochondria of the red blood cell as well as the cytosol, and then back to the mitochondria where the heme is ultimately created, and then it is sent back into the cytosol. From here, each heme molecule will bind with a protein chain called globulin, and then in the red blood cell cytosol, and then this will be this process is taking place in the cytosol. 4 hemoglobin subunits will ultimately come together to create one hemoglobin molecule. From here, the hemoglobin molecule will start being actively used. So hemoglobin is really dominates a red blood cell. So what I mean by that is, like, mature red blood cells will actually shed crucial organelles that you'll find in standard cells, like a nucleus and a mitochondria, allowing more hemoglobin to be packed in to each red blood cell. So in the standard red blood cell, there's up to 280 million molecules of hemoglobin. Because hemoglobin is a molecule within the red blood cell cytosol, specific movement of hemoglobin is largely random, meaning it's in this fluid. It's, each molecule of hemoglobin is in the cytosol, and it's moving around, and it's usually measured. Hemoglobin is mostly measured using like kinetic energy or oxygen saturation, which can actually describe how hemoglobin are acting as they facilitate oxygen transportation throughout the body. During the hemoglobin oxygenation process, oxygen will diffuse from the alveolar sacs within the lungs into pulmonary capillaries, where they will then basically transfuse through the red blood cell membrane and bind with hemoglobin. Hemoglobin will hold on to these molecules and then circulate throughout the body. Once hemoglobin reaches a tissue in need of oxygen, this is like tissues that are experiencing a very high metabolic rate, where the partial pressure of oxygen is like relatively low. This will cause the hemoglobin molecule to expel the oxygen from the heme group and allow that oxygen molecule to be used and diffused into like a muscle cell, where it can be used in the mitochondria to produce ATP and energy in the cell. After a hemoglobin molecule loses O2, oxygen, it's usually rebonded with hydrogen ions or CO2, and then it will transport those molecules back to the lungs to be expelled from the body. So red blood cells typically have a lifespan of about 120 days. After this time, they are removed from circulation via uptake by macrophages of the reticuloendothelial systems in the liver and spleen. Once engulfed in these macrophages, globulin is recycled into amino acids, which is then used for other processes in the body, like the citric acid cycle or fat oxidation. Heme, however, is oxidized, and the heme ring is actually opened by heme oxygenase. This produces a molecule called tetrapyrrole biliverdin. It also creates iron, ion, and CO. Biliverdin, however, is then actually turned into bilirubin. This is actually transported into the liver and then excreted as bile. This bilirubin will travel to the kidneys and then be excreted in the urine, or travel to the large intestines and then be excreted as feces. Okay, that was my application activity. Thanks.